Greetings, parasite ecologists. Today, I want to tell you about some research that my lab is conducting, research that we are really excited about. We're interested to know, is the world wormier than it used to be? And by this somewhat flippant question, I mean, are we living with more or fewer parasites? Are parasite burdens on the rise in this changing world of ours? Given how consequential parasites are both as agents of disease and as threads in the fabric of ecosystems, it's really important for us to know whether they are ascendant or disappearing. A new subdiscipline is now opening up an avenue for us to answer that question: the historical ecology of parasitism. Today, I'll introduce you to that subdiscipline and I'll explain what it has to teach us about the trajectory of parasite populations through time. Along the way, I'm going to show you data from several projects, and I absolutely am not doing these projects alone. I want to acknowledge my fabulous collaborators. This work was supported by the Alfred P. Sloan Foundation, the UW Royalty Research Fund, a UW Innovation Award, and a Career Award from the U.S. National Science Foundation. I also want to make sure to give my heartfelt gratitude to curators at the 14 museums who allowed us to work on their specimens. Ecologists have been obsessively documenting the decline of biodiversity since our field was invented. There is nothing we love more than counting things as they nosedive into oblivion. Take, for example, insects, for whom biodiversity has been in freefall as the results of land use change and the intensive use of insecticidal agrochemicals. Or the plight of amphibians, which have been decimated by a global epizootic of chytridiomycosis, a fungal infection. Birds aren't faring well at all, primarily due to habitat loss, and many mammal species are on the fast track to extinction. We've done a really good job of tallying all of this up for so many species. And of course, everyone cares about the plight of panda bears and pretty birds and even adorable frogs, and we've even gone so far as to do these tallies for icky things, like insects. But parasites? Beyond the pale. We've literally not the foggiest clue about how populations of wildlife parasites have been changing over time. And I think that this is for two reasons. One, they're gross, and no one cares about them, with the exception of us, of course. And there's very little incidental information passed down to us from decades ago, because there just aren't a lot of circumstances where people were recording parasite data for non-ecological purposes in the past. There are fisheries records going back hundreds of years, so people working on fish often have a resource that they can rely on, but no such thing exists for parasites. But it's really important that we figure out what parasites have been up to over the past several decades. Why? Well, for one thing, it sure seems like we've been seeing more outbreaks of parasites recently than we did in the distant past. Think about something like sea star wasting disease, which turned sea stars into piles of mush in 2013 and drove one species, the sunflower star Pycnopodia, to the brink of extinction. If parasites are increasing in abundance, we need to know so that we can anticipate events like this one. On the other hand, we might want to be on the lookout for parasite declines, because parasites are species just like any others, and they may be nosediving, just like insects, amphibians, birds, and mammals. In fact, parasites are, by conservative estimates, 40% of all animal species, and you know that isn't just because there are radiations of parasites that produced hundreds of thousands of nearly identical species. There's a tremendous amount of diversity in the parasites, because parasitism has evolved 223 times just among the metazoa. When we talk about parasites, we are talking about an enormous chunk of Earth's animal biodiversity, absolutely dwarfing the biodiversity of the amphibians, birds, and mammals. But parasites aren't just diverse, they're also critically important components of ecosystems. Consider Carpinteria Salt Marsh, just south of Santa Barbara. This tiny marsh used to be a parking lot, and it was restored a couple of years ago. If you go into this marsh and weigh and measure everything, as Armand Kuris and his colleagues did in the early 2000s, you'll find that the parasites in this marsh weigh as much as a small herd of elephants. Now, no ecologist in their right mind would think that putting a herd of elephants in Carpinteria salt marsh would have zero ecological effect. These parasites are as powerful as a herd of elephants, but because they're hidden inside of their hosts, that power is quite easy to overlook. Parasites are ecologically important for better or for worse, and yet we have literally no clue what their trajectory through time might be. And my goal is to fix that. My lab is dedicated to answering this question, and today I'll tell you about some of the hypotheses we've been interested in testing and how a new subfield, the historical ecology of parasitism, is helping us to do that. Finally, I'll give you some data that weigh in on this central question, at least for a tiny corner of the world, Puget Sound, which is our home ecosystem up here in Seattle. Let's begin at the beginning. There are a lot of reasons to think that things have changed substantially for parasites in the past few decades. We can hypothesize a whole variety of changes for parasites driven by physical, chemical, and biological changes to the biosphere. Here, I'm going to build you a path diagram where positive associations are shown in black and negative associations are shown in gray. Like in any path diagram, you're going to multiply the signs of the paths along a compound path to get the net indirect effect of one node on another. Now imagine something as simple as the increasing temperatures associated with climate change. These could increase parasite transmission by speeding up parasite life cycles, or they could push parasites outside of their thermal tolerance limits. Increasing temps could reduce host immunity, thereby allowing parasites to colonize hosts more easily, or increasing temps could increase the abundance of weedy intermediate hosts who are good at transmission. Or they could decrease the abundance of vulnerable hosts that parasites depend on. A whole variety of net outcomes on parasitism are possible from just this one physical change of increasing temperature. Same goes for habitat fragmentation, habitat simplification, and inputs of nutrient or toxic pollutants. We've got plenty of these fine-scale mechanistic hypotheses, but they all point in different directions, and we have no idea which are the most important for governing overall parasite transmission. What is the net effect across all of these many drivers? When all of these competing forces interact in nature, do they tend to produce increases or decreases in parasite abundance over time? Because we have no long-term data, we currently don't know the answer to that question. But we do have some hypotheses. Many papers have now advanced the hypothesis that parasites may be vulnerable to global change, particularly to biodiversity loss, because they have double the habitat requirements of free-living species. Just like free-living species, parasites have their own niche, the environmental parameters within which they can exist. But unlike free-living species, they also need to overlap with the niche of their host. In a changing world, that double habitat requirement becomes a double vulnerability because the second the host disappears, or even just stops overlapping with the parasite's niche, that's game over for the parasite. And in fact, many of these papers posit that those wildlife parasites with complex life cycles are especially vulnerable, and that the more complex the life cycle, the greater the vulnerability. Just to make sure we keep our terminology straight, remember that we sort parasites into two broad transmission strategies: directly transmitted and complex life cycle parasites. Directly transmitted parasites are passed from one host to another host of the same species. But complex life cycle parasites proceed through a sequence of hosts from one host species to an entirely different species, and they can't skip steps in that life cycle. And that is what might make them particularly vulnerable to environmental change. Here's one way to think about it. Rube Goldberg was an American cartoonist and inventor who lived from 1883 to 1970, and he is famous for creating these chain reaction contraptions that perform a really simple task in an overly elaborate way. These machines have always reminded me a little bit of the complex life cycles of parasites. because you can take something as simple as sexual reproduction, and then you add host species and larval stages and other hoops to jump through just to get back to the good part. These machines are a really effective way to think about the vulnerability of complex life cycles in a changing world. Imagine. Each mechanism in this machine is just one stage of a parasite's complex life cycle, and each mechanism depends on the previous one. If one mechanism breaks, the whole machine stops. And the thing about Rube Goldberg machines is the more mechanisms you have, the greater the likelihood that something is going to go wrong. Now think about parasite life cycles. As the world changes, it's inevitable that mechanisms will break, that obligately required hosts will diminish in abundance, or their timing will change in a way that doesn't line up for their parasite anymore. In a changing world, it's actually only a matter of time before one mechanism breaks, and the more mechanisms you have, the likelier that is to happen and the faster it's going to happen. Complex life cycle parasites are marvels of nature, unparalleled in their complexity by any free-living groups. But these complicated, elaborate, delicate, and precarious life cycles may be easily broken by environmental change. And that's why, folks argue, parasites with more complex life cycles will tend to be vulnerable in a changing world and increasingly less likely to be able to turn the page. So here we have these very clear predictions, but literally zero data with which to test those predictions. And that is where the historical ecology of parasitism comes in. Historical ecology is a pretty young field. Until now, it's been entirely focused on free-living species, and it's made some really great strides in the past couple of years. Historical ecologists use data sources that were not originally collected for ecological purposes to draw inferences about the past presence or abundance of species. And people have come up with some really creative ways to glean this information, particularly for historical assemblages of fishes. Some have used art, like this Roman mosaic showing a dusky grouper, a species which is now endangered and very rare in the Mediterranean. Others have used the composition of species represented in seafood menus to learn about change in fish assemblages over time. And of course, people like to take pictures of the big fish that they catch, and creative folks have extracted from those photos qualitative and sometimes even quantitative data to contrast against modern fish assemblages. And these pictures can be really striking, bringing home the fact that ecosystems look very different today than they did even just a couple of decades ago. Now, parasites are just not that charismatic. No one takes pictures of them while they're on vacation. You're not going to find them on a menu, and not many works of art are devoted to them. And so historical information on parasite assemblages is harder to come by, unless you consider one source: museum specimens. When animals are dunked into a jar of ethanol or formalin, their parasites are preserved right alongside them. Now, say you find yourself in a natural history museum anywhere in the world. In the public spaces, you're going to find many specimens, but these are just going to be the biggest, the rarest, the most beautiful. Meanwhile, if you hop in an elevator and take it down into the bowels of the museum to areas that are closed to the public, there are millions of jars held in much less attractive conditions. And I'm talking basements and sub-basements full of jars stretching from the 1700s to the present day. And each one of the animals preserved in these jars is a parasite time capsule. If you perform parasitological dissections on these animals, if you open up these parasite time capsules, you can find tons of parasites hidden within. And my lab's research, as I'll show you in just a moment, suggests that these parasites are just as well preserved in fixed hosts as we see in fresh specimens. Just as easy to find, just as easy to count. And that means that we can pick jars to create a timeline for a given host species, and we can see how that host's parasite burden has ebbed and flowed through time. This is the historical ecology of parasitism, and this is how we can get data on parasites of the past. despite the fact that no one has historically cared enough to track parasites. But when my lab group started in on this line of investigation, the first thing that we wanted to know was, can you get reliable quantitative information on parasite burdens from natural history specimens? Do the parasite counts that you get out of a natural history specimen reflect what is actually in that individual or population in nature? Or does something about the preservation process bias parasite counts? To answer that question, we performed an experiment. Master's student Evan Fiorenza got a hold of a bunch of fish and he randomly assigned them to either be experimentally preserved with the museum's preservation protocol, or they were put into a control group where nothing was done to them. We focus on fish because they're easy. They're plentiful in museum collections, and many of the parasites in aquatic ecosystems pass through fish at some stage of their life cycle, so they give us a nice cross-section of the full parasite community. But this could be done with lots of other vertebrate and invertebrate hosts. I just want to mention that most of our work to date is on fish. Anyway, so Evan had these two groups of fish. He performed parasitological dissections, and he looked for differences between these two treatments. And what Evan found was that there was a minimal influence of preservation on parasite detectability. He found 27 parasite species, and of these, only 3 differed between the control and the preservation treatments. Evan aggregated the response to preservation across all of those 27 parasite species, and he found that no group was easier to detect in one treatment versus the other. Values to the right of this axis indicate parasites that were easier to detect in the preservation treatment, and on the left, they were easier to detect in the control treatment. There's no difference between the treatments when you look across all parasites, just at the larvae versus the adults in blue, or at the various taxonomic groupings of parasites in rainbow colors. Okay, with these data, we can be pretty confident that the museum specimens are providing us with reliable information on parasite abundance. And armed with that information, we now knew that we could use museum specimens to reconstruct really long time series of parasite abundance. Now let's dive in and use this new tool to answer the question, is the world wormier than it used to be? First, let's fancy up the question a little bit. What we're really interested in is how and why has parasite abundance changed over the past century? To tackle this problem, my collaborators and I launched a project focused on Puget Sound, which is a big inlet of the Pacific Ocean, the second largest estuary complex in the United States. We picked it because home seemed like a good place to start. We also picked it because our university's museum, the Burke Museum of Natural History and Culture, has outstanding representation from the Puget Sound region. We were able to get enough specimens to evaluate change over time in the parasite burden of 8 different host species. And these fish species were chosen to span a variety of trophic levels and body sizes so that we would maximize the diversity of parasites that we would find in them. Most of the specimens were available at the UW fish collection, although we supplemented from 13 other collections as well. For many of these species, we were able to get back into the 1880s. We were really careful in our selection of specimens for dissection. We got a good mix of specimens from across Puget Sound and across time, with minimal correlation between geography and time. On the map at left, you'll see each specimen plotted with older specimens in darker colors. Notice the nice mixed distribution of colors across space. Another way to visualize this is to just take away longitude entirely and plot the latitudes of the specimens across time. Here, time is on the x-axis, latitude is on the y, and the different colors represent different host species. And you can see the colors are really well mixed, meaning there's little correlation between species, location, and time. And you can also see that we've got really good replication from about 1920 to 2019. It's a good thing, too, because a lot was changing in Puget Sound over that time period. Here you'll see time on the x-axis and the magnitude of a variety of potential environmental drivers of parasite burden on the y. Now remember, we have a bunch of hypotheses for how these various drivers are going to affect parasite abundance. Temperature has fluctuated through time, but increased substantially in the past few decades. This band is going to get thicker and darker where temperature is higher. These data on annual sea surface temperature of Puget Sound are from a continuously maintained record at one of our local lighthouses dating back to the 1920s. We had access to data on 12 different pollutants, and we reduced the dimensionality of that data set to two principal components, one of which is driven by nutrient pollution, and that one peaked in the 1960s, just before the implementation of the U.S. Clean Water Act. A second principal component is driven primarily by toxic contaminants like copper. And then we also have the density of 6 of our 8 total host species from data that my collaborators, Tim Essington and Corey Green, were able to assemble. Remember, we're trying to understand both how and why parasite abundance has changed over the past century, and these potential environmental drivers are going to help us get at that why question. Neither of these questions would have been possible to answer if we hadn't gone to the trouble to assemble a truly massive data set. In total, we counted 17,702 parasite individuals in 699 hosts. Our time series stretches from 1880 to 2019, but the vast majority of the data are from 1920 to 2019. Our model wouldn't converge with the super rare parasites included, so we narrowed ourselves down to parasites that occurred at least 5% prevalence in their host species. And that gave us 85 parasite taxa, and among those 85 parasite taxa were 17,259 parasite individuals, making this dataset the largest long-term dataset of wildlife parasite abundance ever assembled. And it's a good thing, too, because we wouldn't have been able to detect patterns without the high level of replication we achieved across parasite taxa, host taxa, and time. Now, one last thing before I show you some results. Remember our Rube Goldberg machine? We wanted to test the specific prediction from the literature that greater life cycle complexity would make parasites more vulnerable to decline. And so we made sure to include that in our models. This is a big, scary graph, so let me break it down for you. We're going to be looking at trajectories of parasite abundance change for individual parasite species. On the y-axis, we have each of our 85 parasite species or taxa. They are organized by the kind of life cycle they have. Some parasites have just one obligately required host in the life cycle. Remember, we call these directly transmitted parasites, and we had 20 of those taxa. Other parasites need 2 different host species to complete their life cycles, and some require at least 3. I'm going to call any parasite that requires more than one host a complex life cycle parasite. We had 21 parasite taxa that obligately require 2 hosts, and 44 parasite taxa that obligately require 3 or more hosts. The x-axis is the random effect of year for each parasite taxon, so parasite taxa that are positive on this x-axis are increasing through time, and ones that are negative are declining through time. And with that, let's take a step back. We see some parasite taxa making major gains through time, and some experiencing major losses. About 40% of all the parasite taxa have a significant time effect in either the positive or the negative direction. But if you look at the groupings, you're going to find about even responses, positive and negative, among the one-host parasites, about even responses among the 2-host parasites, and a tendency toward decline among those parasites with 3 or more hosts in their life cycle. If we generate a prediction plot for each of these groups that summarizes over this variability, this is what it looks like. Directly transmitted parasites and complex life cycle parasites with only 2 hosts don't change over time. But parasites with 3 or more hosts in their life cycle plummet. The scale of this decline is pretty enormous. It's 11% per decade, which might not sound like a lot, but it's actually greater than the magnitude of decline that's triggered conservation action for things that people actually care about, like mammals and birds. This is a massive, previously undetected loss of parasite biodiversity. But we not only wanted to know how parasite abundance had changed over the past century, we also wanted to know why. And we had this fantastic data set of environmental parameters that line up with the time series of our parasite data. We were able to build some statistical models to find which of these environmental variables was most closely correlated with the declines that we observed in complex life cycle parasites through time. We limited this analysis to just the parasites with 3 or more host species in their life cycle, since that is the group of parasites that is plummeting in abundance and whose change over time we want to explain. Let's take a look at the slope of the relationship between each potential driver and parasite abundance. On the x-axis, we have year, temperature, the 2 principal components for pollution, and host density. We already know from the previous analysis that the relationship between time and the abundance of 3 host parasites is negative. Not telling you anything new here. We found that the 2 pollutant principal components and host density were not associated with parasite abundance. But sea surface temperature was associated with parasite abundance. And the magnitude of the effect was very similar to the effect of year. So we believe that the decline that we observed in parasites over time is correlated with temperature increases driven by climate change. When we look at parasite abundance as a function of sea surface temperature, we see that parasite abundance declined with increasing sea surface temperature at a rate of 38% for every 1 degree C increase in temperature. 1 degree Celsius is a little less than the amount of warming that Puget Sound has seen since 1950. So what's the upshot? First of all, the loss of parasite biodiversity that we documented is massive. Puget Sound has lost 11% of its 3 host parasites per decade, and that makes it a lot less wormy than it used to be. And of course, this is just a correlational study, so we can't be definitive about causes. But of the potential drivers that we screened, climate change is the most likely culprit. Sea surface temperature is, of course, positively correlated with time, and the true driver could be something else that is also correlated with time. But at the moment, climate change is the prime suspect. How might climate be messing with complex life cycles? Well, the more hosts you have, the more potential points of failure. Climate change produces winners and losers, and in a complex life cycle, if you lose any one host, it doesn't matter if other hosts are winning. It's still game over for you. Adding hosts to the life cycle might make the complex life cycle parasites particularly vulnerable to any kind of change. These parasite life cycles evolved in periods of comparative stasis. They are not prepared for the rapid shifts that are occurring due to climate change. Compared to other parasites that live more simply, there are just too many ways for things to go wrong for complex life cycle parasites in a warming world. And what appears to matter is not necessarily the trajectory of the host in which the parasites were counted in this study. Host density, fish host density, didn't explain any significant amount of the variance in our models. Where the Rube Goldberg machine is breaking down, I suspect, is at other points in the life cycle, potentially among the invertebrate intermediate hosts or the high trophic level definitive hosts. So, Puget Sound is less wormy than it used to be. What does that mean? Well, let me start out by saying that this dataset is really exciting. It's our first tiny peek into what wildlife parasites have been up to over the past century. More than half of the parasites that we found were complex life cycle parasites with 3 or more hosts in their life cycles. And these parasites are going down with the ship. 1 or 2 species are escaping on lifeboats, clutching bags of stolen silverware, but a huge chunk of parasite biodiversity is being dragged down into an icy grave. We started off with all of these opposing mechanistic predictions for how the various cross-currents of global change would produce various potential outcomes for parasite abundance in a changing world. But the one thing that our data are most consistent with is this single pathway, where increasing temperatures, interfere with a few vulnerable host species, and that interferes with parasite transmission. Mere observational data, screening across broad swaths of time, space, and taxonomy, lets us figure out what are the overarching patterns, and now we've narrowed in on some specific mechanisms that appear to be important for driving those patterns, and which we can then investigate further in smaller scale, manipulative experiments. Now, I know what question is probably on your lips right now. This is happening in Puget Sound. Is it happening elsewhere? I can't answer that question for you. This dataset right now is unique. We won't know for sure until researchers collect similar kinds of data in other parts of the world. But in the meantime, it's not an encouraging sign. The very first peek we've gotten at the long-term trajectory of parasites is pretty disturbing. It reminds me a lot of the countdowns to extinction that we've done for a bunch of other taxa. But in fact, it may be even worse than that. There are papers in Science and Nature raising alarms about the declines of some other taxa, and they'll find declines of 6.3% per decade, 6.9%, even 8.8%. Meanwhile, we found an 11% decline per decade in some complex life cycle parasites. We're losing parasites, and while that may sound to some folks like a good thing, I'm confident that it isn't. This massive loss of parasite biodiversity has implications for biodiversity writ large, given that animal diversity, at least, is largely parasitic. But beyond the impact on global biodiversity counts, I wonder, what are the ecological impacts of parasite loss? What's Carpinteria salt marsh gonna look like without its herd of elephants? We don't know, but we're rapidly moving into a world where we're gonna find out. It might be time to think about parasite conservation. And I know that that sounds like a contradiction in terms. Parasites are things we're supposed to want to destroy, not things that we're supposed to want to protect. But parasites are species like any others, and at least in Puget Sound, they are nosediving. We'll learn more about the potential consequences of parasite biodiversity loss later on in this video series. In the meantime, this is the historical ecology of parasitism, our way of understanding how parasite populations have changed through time. What do you think about parasite conservation? Let me know in the comments below, and I'll see you in the next video.